Hello, everybody. We have a really large topic ahead of us today, fatty acid synthesis. So let's get started. Our learning goals today will be to outline the conditions needed for fatty acid synthesis, where it happens in the body and the cell, and when. With that in mind, we'll illustrate the mechanism of fatty acid synthesis itself. And then we will be contrasting the regulatory mechanisms of fatty acid synthesis and oxidation. Today's molecule of the moment is actually a class of molecules called the beta keto esters. And perhaps the first method they were formed synthetically was through a condensation of two ethyl acetates, which I'm showing over here. When you add a strong base to a solution of ethyl acetate, usually something like sodium ethoxide, it will combine to form this ethyl acetoacetate, aka a simple beta ketoester. Now, previously in beta oxidation, that's simply a four-step cleavage reaction of this C alpha C beta bond over there. And we learned that in last class. But this class is about fatty acid synthesis. We will observe the formation of this bond. So I'm asking you, how does this condensation work? If it's been a while since you took organic chemistry, I'll give you a hint. The alpha carbon is going to be weakly acidic. Remember that this is the carboxylic acid carbon, and then this is the alpha carbon. The protons on there are subject to deprotonation by this very strong base. So chew on that, and then we'll circle back to this first thing in lecture. Before diving into the nitty-gritty, I'm going to repeat something that I've definitely mentioned before. Anabolism and catabolism are never just the same pathways done backwards. Never. In the case of fatty acid metabolism, fatty acid synthesis isn't just beta oxidation in reverse. And to prove that to you, let's map out some of the global differences between these chemistries. Starting with fatty acid catabolism, aka beta oxidation, it's going to occur steadily in most tissues, but we're going to notice an uptick in the rate of it in cells that present lots of glucagon receptors. In terms of location, this chemistry occurs exclusively within the mitochondrial matrix. And at an organ level, why, yes, it does occur broadly in most tissues, but we see a lot of beta oxidation occurring in liver and muscle tissue. Now let's contrast this with fatty acid anabolism, aka fatty acid synthesis. Where is it upticked? Well, in fed tissues, particularly those with lots of insulin receptors. The process itself is exclusive to the cytosol in animals and the chloroplasts in plants. And indeed, we do lots of fatty acid synthesis in the liver, but also in adipocytes as well. So generally speaking, these processes dominate in different organs in different conditions, and they also occur in different locations within the cell. This foreshadows that fatty acid synthesis is going to employ different enzymes than the ones used in beta oxidation, and therefore these are two separate pathways. We can break fatty acid synthesis down into two general phases. The first phase is called initiation. And here the goal is to collect and activate so-called monomer for condensation into eventual fatty acid. And we have a description of that over here on the right. We're going to sequester acetyl-CoA and activate it with bicarbonate and ATP to form this activated unit malonyl-CoA, which is going to participate in the next step called elongation. And what is elongation about? This is the complicated part, in my opinion. It is going to constitute a 4-part spiral reaction involving monomer addition and reduction. We are going to take these malonyl-CoAs and glue them together and reduce them until we get a C16 palmitic acid, which is usually where the spiral ends. And the core ingredients for this process of initiating and elongating are going to be twofold. We first need a lot of reducing power, and that makes sense because fatty acids are highly reduced molecules, and acetyl-CoA and malonyl-CoA are a little bit more oxidized, so we have to undo that. And what the cell uses as the hydride donor to reduce these substrates is NADPH. That is going to be our hydride source. Take note that we are not using NADH, which is generated in beta oxidation. That's a deliberate difference between these two pathways, and it's going to in part help prevent them from cycling into each other wastefully. The products of one are not the literal reactants of the other. And of course, we're going to need a carbon source as well for fatty acid synthesis, and that is going to be, as mentioned, acetyl-CoA. It really is worth repeating that fatty acid synthesis, again, in a nutshell, is stitching together these acetyl-CoAs and loading them up with hydrides from NADPH. As I just mentioned, fatty acid synthesis begins with acetyl-CoA in the cytosol. There's just one problem. Acetyl-CoA, by design, is trapped inside the mitochondrial matrix. I mean, the citric acid cycle needs it. So we need a special fix to get that acetyl-CoA out of the mitochondria, and we call that the citrate shuttle. And this is how it works. When the cell is well fed and it's rich in NADH and ATP, that is going to prompt the citric acid cycle to shut down. And we are going to see an accumulation of citrate. Now, citrate can cross both mitochondrial membranes thanks to certain citrate transporter proteins. So citrate channels leak excess citrate into the cytosol. And out here in the cytosol, there is another enzyme called citrate lyase. And what that will do is take an equivalent of coenzyme A, it will take an equivalent of ATP, and it's going to convert one molecule of citrate into oxaloacetate and money in the bank, acetyl-CoA. That acetyl-CoA gets directly fed into the elongation reactions of fatty acid synthesis. Oxaloacetate is still useful. We're not done with this yet. We are going to follow that up with additional enzymes, malate dehydrogenase, and most importantly, malic enzyme. And malic enzyme is going to oxidize it and generate an equivalent of NADPH. Remember how we need NADPH to do fatty acid synthesis? This is where 1/2 of the NADPH needed comes from. The other half we get from other chemistry that occurs in this cell. When we're done with this malic enzyme process, we're going to end up with pyruvate, which we can then import back into the mitochondrial matrix and convert into oxaloacetate again with the help of another enzyme, pyruvate carboxylase, which is going to use another equivalent of ATP. So all in all, the net cost of the citrate shuttle is 2 ATP. The second half of the initiation step is activation of the acetyl-CoA. But hold the phone, you might be saying. Coenzyme A is already activating this acetyl group. It's a leaving group, after all. We've seen this before, time and again. So why does acetyl-CoA need to be activated if it's already activated? And the reason is, well, acetyl-CoA is not activated enough. All right? Lipid synthesis is really challenging. It's really chemically uphill. And we're able to cross that high activation barrier by lifting the energy of the reactants further, by upgrading acetyl-CoA into malonyl-CoA. The activity of acetyl-CoA is derived from its thioester group. Malonyl-CoA has got a little bit extra kick to it because it's a beta keto thioester. That beta position is really special, all right? We saw this last time during beta oxidation. Beta ketoacyls slash acids are high energy. And the reason why they're high energy is because we have entropically favorable decarboxylation. This COO minus group is going to pop off as a CO2, which can open the door for establishing a new, longer carbon-carbon chain based off of this position. The entropics of releasing a CO2 is what will drive fatty acid synthesis towards the product direction. Another quick note about malonyl-CoA, it's not only the essential feedstock for the elongation steps coming up next, it will inhibit the carnitine shuttle that we learned about in last lecture. This will prevent fatty acid importation into the matrix. And therefore, it's going to shut down beta oxidation hard. This is why biology designed synthesis and degradation to occur separately. If it didn't, our cells would just make and break fatty acids in a pointless loop. The enzyme that catalyzes this activation of acetyl-CoA into malonyl-CoA is called ACC, or acetyl-CoA carboxylase. And this is a really cool enzyme in my opinion. It's actually two enzymes built into one protein. There's first the biotin carboxylase domain, and then second, a trans carboxylase domain. And the cofactor that is working between these two sectors is everyone's favorite, biotin. And we've seen biotin in action before. I want you to think of biotin as biology's master CO2 wrangler. The mechanism can be boiled down into the following steps. First, we're going to see biotin carboxylase do its thing. And the first thing it's going to do is use an ATP molecule to activate a dissolved bicarbonate ion, which you can think of as dissolved CO2. And it's going to activate it and form something called carboxyphosphate, which looks like this. We've now installed a CO2 molecule with a good leaving group, the phosphate. It's going to make our carbon rather electrophilic. So the nucleophilic biotin can swoop in and attack and bind it. That's going to be the next phase of biotin carboxylase. We are going to condense carboxyphosphate onto biotin, and we will get the following adduct highlighted over there. Then biotin is going to function as a swinging arm. It's going to rotate over to the trans-carboxylase domain, where it's going to take this activated CO2 group and it will slam it onto acetyl-CoA, forming malonyl-CoA in the process. The precise mechanism of this is poorly understood. We do suspect it involves deprotonating that methyl group on acetyl-CoA into an enolate, which will then attack the CO2 group on the biotin. All I expect you to know, though, is how we got this CO2 group installed on our acetyl-CoA. We started with CO2, from the air. It got into our cells and it dissolved into the bicarbonate ion. We merged it with a phosphate from ATP, which put it on biotin, and then biotin inserts it right over there. Bam, malonyl-CoA. The second half of fatty acid synthesis, aka elongation, will be the main point of fare from here on out. And the good news is that a single enzyme called fatty acid synthase 1 is responsible for connecting these malonyl-CoAs together into a single fatty acid. The bad news is that fatty acid synthase is actually a homodimer of two humongous polypeptides, And each polypeptide sequence is going to contain 7 different catalytic sites. This is basically 7 enzymes baked into one protein. They are in order of operation MET, which stands for malonyl-CoA ACP transferase. ACP, that stands for acyl carrier protein. KS, beta ketoacyl ACP synthase. KR, beta ketoacyl ACP reductase. DH, beta hydroxyacyl ACP dehydratase. ER, anoyl ACP reductase. And it's not shown in this graphic, but connected to the ACP domain is the final enzyme, TE, which stands for thioesterase. I'm going to pause briefly and say that there may be some familiar sounding terminology present. Terms like enoyl, beta hydroxyacyl, beta ketoacyl. Where have we heard these words before? These words were in the names of the intermediates present in the fatty acid spiral, which we learned last time. So this hopefully alludes to the fact that the chemistry that occurs during fatty acid synthesis and fatty acid breakdown does indeed have some similarities. You should also know that there are 2 sites where covalent catalysis occurs in fatty acid synthesis. First, we have KS, which bears a very special cysteine residue. And its job is to form a thioester with either the growing fatty acid chain or special acetyl group. We'll talk more about that soon. And secondly, ACP is going to possess a cofactor that resembles coenzyme A called 4-phosphopentythene. This cofactor will bind either malonyl-CoA or the growing fatty acid chain, depending on what step we're on in the mechanism. And I know this all sounds really overwhelming right now, but we're going to dive in and dissect each portion individually soon enough. I just want to dedicate some time to the big picture before we dive into the forest. Before we get into the weeds, let's do a brief tour on how these moving parts all work together. Like the fatty acid spiral, fatty acid synthesis will proceed via 2 carbon unit additions. Now, malonyl-CoA, you may recall, has 3 carbons in it. 1, 2, and 3. That over here is malonyl-CoA attached to the ACP portion of fatty acid synthase. What elongation does is we're going to drive off the COO portion that we just installed via the initiation enzyme ACC, release it as this green colored CO2, and that's going to entropically drive this really difficult addition of this orange unit right over here to this special acetyl unit held in reserve on the KS domain of this enzyme. And that is going to move this yellow block down the line to accommodate the insertion of this red activated portion of malonyl-CoA. Also, take note of what happened to the C double bond O that was in the yellow block. If we move over here, we notice that the C double bond O has now been replaced with CH2. We reduced it. Beta oxidation is highly oxidative. The fatty acid synthesis machinery is highly reductive. each spiral is going to consume 4 electrons and 4 protons because we need to get rid of all of these C double bond O's present. I also want you to take note that as we add on each 2-carbon unit, we're actually building from the omega carbon to the alpha carbon. Take a look at this graphic over here again. Chain growth begins with this yellow acetyl. Then we add in this orangish acetyl from the ACP domain right underneath it by driving off a CO2. And then we're going to repeat the process 3, 4, 5 times and so on, starting with then with the blue one, which gets put over there. And then another blue 4th one. And then we're going to repeat this until we have a C16 fatty acid, palmitic acid. At this point, the substrate has grown too big and unwieldy for the fatty acid synthase machinery, and then we're going to cut it off with TE, thioesterase. But this is fatty acid synthase in a nutshell. We are going to load up fatty acid synthase. We are going to remove CO2, which will let us staple together malonyl onto the growing chain. And then we're going to reduce. And then we repeat. Before we can even begin the elongation cycle proper, we need to preload the fatty acid synthase complex with those 2 covalent intermediates we discussed 2 slides back. We need the acetyl-CoA primer on the Ks domain, and we also need malonyl-CoA on ACP. And we need a few of these subdomains working together to achieve this goal. And part 1 of this priming step is going to involve MAT, malonyl acetyl-CoA ACP transferase. The job of this enzyme is to load stuff onto ACP, acyl carrier protein. So let's begin with the acetyl-CoA primer. We are going to have Matt grab the acetyl-CoA and prompt it to form a thioester bond onto ACP, specifically the 4-phosphopentothene coenzyme. So now that Matt was able to transfer acetyl-CoA onto ACP, ACP is going to carry it to the Ks domain. ACP is kind of like lipoamide or biotin. It's going to be a flexible arm that's going to reach over and deposit the acetyl fragment it picked up onto that cysteine. And then it's going to come back to the starting position next to Matt, and it's going to get loaded up again, this time with malonyl-CoA. It's going to work together with Matt, and then bam, we have a second thioester formed, this time between our second carbon units. And remember that these CoA groups, they're ultimately going to be leaving groups, so we're going to lose both of them as free. We call these CoAsh, the free sulfhydryl, and that's going to be released when each of these gets attached onto our protein covalently. So to recap, we are going to have Matt take an acetyl-CoA, stick it onto ACP, and ACP will stick it onto Ks. And then we will have Matt take malonyl-CoA, stick it onto ACP, and then stop. Now we are ready to do our first step in the elongation cycle. Now we officially begin the elongation cycle. ACP, which we just loaded up with malonyl-CoA, it's got 3 carbons, 1, 2, and 3. It's going to swing over to KS's active site, where it's going to react with that acetyl group we left primed on KS in the previous step. And the malonyl-CoA and the acetyl-CoA that we just added on are going to react together through a process known as decarboxylative condensation. What does that mean in plain English? This CO2 unit that ACC installed during activation is going to come off. It's going to leave as a carbon dioxide molecule through an electronic rearrangement. The electrons are going to flow this way, and it's going to prompt the cleavage of this C alpha, C beta bond. And the cleavage of that bond is going to help form a new C alpha and C beta bond with the acetyl that's left on the KS domain. That's the new beta carbon that's going to ultimately take the place of that green CO2 unit. And while this is all happening, don't forget that the CO2 is serving as a leaving group, and hence decarboxylative condensation. Removing the CO2 is going to drive the formation of a new carbon-carbon bond. You may have learned this reaction in organic chemistry. We call this a Claisen condensation, if you recall. And remind me, what are the conditions that we need to perform a Claisen condensation? We need a strong base. Well, we can't do that in biology. We can't deprotonate the alpha carbon and turn it into a powerful nucleophile like we're used to, because a strong base is going to be toxic. It's going to destroy the cell. So instead, we form the enolate, so to speak, by this decarboxylation process. This decarboxylation is a cheat code that allows us to condense this alpha carbon onto this beta carbon without using a strong base. By attaching the reddish orangish acetyl fragments that came from malonyl-CoA onto the yellow acetyl fragments that began on KS and is now attached to ACP, we have just generated a ketone in the beta position to this thioester. Which means we have a beta ketoacyl. That ketone in the beta position needs to go. Our goal is to remove via a reduction the C double bond O so we can saturate our growing baby fatty acid. And to do that, ACP is going to carry this growing fragment to a different domain, the beta ketoacyl ACP reductase. And just as the name describes, it is going to reduce the beta ketoacyl. And what happens when you reduce a beta ketone? You get a beta alcohol. So the product of reducing this compound is D-beta hydroxybutyryl ACP. And now we have a hydroxy group in that beta position. So what is the reductant employed in this reaction? Well, it's NADPH. NADH is catabolic. NADPH is anabolic. So this molecule is going to be an electron donor for reactions that build up compounds. And remember, we get NADPH in part from malic enzyme, which we observed in the citrate shuttle. That is going to provide about half of the NADPH required for the formation of these fatty acids. The other half comes from another reaction pathway that we haven't discussed called the pentose phosphate pathway, which is also really good at making NADPH. Step 3 of elongation is a dehydration reaction. ACP will deliver the beta alcohol that we just made to the dehydratase domain, DH for short, where it is going to dehydrate that alcohol and release it as a water molecule. When you dehydrate an alcohol, you essentially perform an elimination reaction, and that's going to yield a double bond. We will now desaturate the C alpha C beta bond we just formed into a trans alkene. Trans delta 2 butanoyl ACP is the product of this reaction. We have now fully removed that oxygen from the beta position. All we have to do now is break and hydrogenate that double bond, and we will have effectively converted the yellow portion of this growing fatty acid into a saturated hydrocarbon. ACP is now going to hop on down to the last domain that we haven't yet visited, enoyl ACP reductase, ER for short. And it's going to do just what the name says. It is going to reduce the enoyl group into an alkane, specifically into butyryl ACP. And we see that over here. We start with a double bond, and we end with a single bond connecting the yellow and red-orange fragments. And going from an alkene to an alkane is a reductive process. We need an electron donor, and that donor is going to, of course, be, yet again, NADPH. This process will consume the second NADPH required in each spiral. Now, technically, butyryl-CoA can be hydrolyzed right here and now and liberate what's called a short-chain fatty acid, butyric acid. But biology prefers to keep this process going for a few more additions and get the most bang out of our buck and make a decently sized fatty acid for all this effort. In step 5, ACP will transfer this butyryl group that we just made back to KS where we began in preparation for the next round of elongation. We call this acyl transfer. And what we're essentially doing is repeating step 0 in a sense, except our baby fatty acid chain is taking the place of that first acyl group primer that was put onto KS for the first spiral. So ACP is going to swing back to KS. We will have a trans-thioesterification occur and deliver this growing chain onto that cysteine, which will free up ACP, which can just go down to the next door neighbor, Matt, and then pick up another malonyl-CoA unit. And once we do that, we can repeat the spiral. We can swing back and decarboxylate to form the beta ketone, shown over there. Then we reduce the beta ketone into an alcohol. Then we dehydrate that alcohol into an alkene. And then we saturate that alkene into an alkane. Return it back to the Ks domain. Pick up another malonyl. Rinse and repeat. Attach to form the beta ketone. Reduce to an alcohol. Dehydrate the alcohol. And then saturate the alkene to an alkane. And then you keep on going until it's time to stop. I will repeat again that the elongation cycle is going to flip through the following 4 steps of condensing into the ketone, followed by reducing it to an alcohol, dehydrating it into an alkene, and then reducing that into an alkane. And that will be iterated so many times until we obtain a C16 saturated fatty acid, aka palmitic acid. And that's essentially going to be 7 elongation spirals in total. And the reason why we iterate until we get to the C16 level is because the thioesterase domain, TE, which wasn't shown in those cartoons in the previous couple of slides, that's actually going to best recognize palmitic acid and hydrolyze it off of the fatty acid synthase machinery only when we grow out to that size. And so in total, what does this whole process cost us? The initiation steps is going to consume 7 acetyl-CoAs, 7 carbon dioxides, and 7 ATP molecules, which is going to produce our activated malonyl-CoA, 7 of them, along with 7 equivalents each of the waste products from ATP hydrolysis. We are going to feed in the malonyl-CoA to our elongation reactions, which is going to consume 1 more acetyl-CoA as part of the primer, 14 equivalents of NADPH plus protons, and all of that is going to give us our C16 palmitate, 7 CO2s as waste, 8 coenzyme A's, and 14 spent NADP pluses. We also generate a decent amount of water. We get 6 equivalents. Technically, we make 7, but we're going to exploit and use one of those waters to hydrolyze off the product from FAS via the activity of thioesterase. So that's why the yield is 6 waters and not 7. I will also state the obvious that fatty acid synthesis is metabolically expensive. This is a lot of inputs we're investing into one lipid molecule. It's going to consume 1 ATP per spiral and 2 NADPHs per elongation spiral. And don't forget the cost of the citrate shuttle that we had to employ to get the acetyl-CoA into the cytosol. The citrate shuttle is worth 2 ATPs for citrate, which gets converted to acetyl-CoA. Fatty acid synthesis and oxidation are both tightly regulated to ensure that bodily fuel is stored and released wisely. Of this whole process, ACC is the rate limiting step. This is the reaction that generates malonyl-CoA and is therefore right before the elongation cycle. When active, ACC is going to form these polymers large enough that can be observed under electron microscopy, which is pretty cool. And its activity is controlled at the effector level, phosphorylation level, and transcriptional level. Starting with the small molecules, citrate itself is going to be an activator of ACC. And that's an example of feed forward stimulation. It's actually going to help bundle ACC into these catalytic filaments. And this makes sense because when fuel is abundant, citrate's going to be abundant, and it's going to not only help shut down glycolysis via PFK, it's going to ramp up fatty acid synthesis at the same stroke. Conversely, palmitoyl-CoA, when its concentration rises, that's going to be a negative effector, and it's going to disassemble the ACC filaments. This is an example of product inhibition or negative feedback. Making lipids, after all, requires a lot of energy. If the body doesn't have the energy to ship off these fatty acids elsewhere, it makes sense to slow things down. The phosphorylation state of ACC itself is directly toggled via hormones too. Glucagon and epinephrine will trigger an AMP cascade, and the product of that cascade is going to be phosphorylation. This will have the effect of inactivating fatty acid synthesis because that's going to increase the negative charge on these ACC monomers, and it's going to cause the filaments to fall apart, deactivate. Likewise, insulin, that is going to also stimulate a cascade, which is going to result, instead of a kinase being activated, going to activate a phosphatase, which results in dephosphorylation. And therefore, activation. Insulin gets released when there's plenty of chemical energy, flooding our cells, which means we have the fuels and resources needed to build these up into long-lasting lipid forms of chemical energy. And lastly, at the transcriptional level, we have something called, it's a bit of a mouthful, carbohydrate responsive element binding protein, which we call CHREBP for short. And this is a transcription factor that is sharply spiked during high caloric intake, kind of like insulin, and it's going to be sharply decreased during fasting. Consequently, that means the concentration of ACC itself is going to increase when eating. And when you're not eating, the concentration of this enzyme will go down. And don't forget to take 10 huge steps backwards and synthesize how fatty acid synthesis pairs with the rest of metabolism. When fatty acid synthesis goes up, we are going to have a lot of malonyl-CoA. And that is going to inhibit the carnitine shuttle required for beta oxidation. So that's going to cause fatty acid catabolism to go down. This is another example of reciprocal control. We can't have both of these competing processes dominating at the same time in a given cell. Then take another 10 steps backwards and think about glycogen metabolism. Glycogen metabolism is also regulated through phosphorylation, let's say. Ask yourselves, what does net phosphorylation do to fatty acid synthesis? And then also compare that to what net cellular phosphorylation does to glycogen synthesis. Same for the dephosphorylation. When we dephosphorylate within the cell, that's going to have an activating effect on fatty acid synthesis. Predict if you think that's going to activate or deactivate glycogen synthesis or glycogen breakdown. I recommend after you wrap this up, flip back to your glycogen notes and see how these two forms of metabolism also work together. That is the tricky but beautiful thing about biochemistry. Each of these cogs don't turn in isolation. It is all interconnected.